Hi everyone, welcome back, and we're here for our final Hudson Canyon Live. I'm Kate Clemmer, and I'm coming to you live directly from Hudson Canyon. We're super excited to be here today. We have a lot to share with you, a lot of discoveries that unfolded just this morning. We can't wait to take you down to the lab later today and learn from some of our scientists and experts there as well. So again, this is going to be our expedition highlights. We're going to run through all the different things that we've seen and just share the memories that we've made along the way over these 2 weeks out here in Hudson Canyon. Another special thank you to NOAA Fisheries, the Office of Marine Aviation Operations, the Ocean Exploration Cooperative Institute, and the Interspace Center at the University of Rhode Island. So a lot of this that we're doing out here today, it all can't happen, science at sea can't happen without the help of a ship crew. So we're actually going to hear from some of our operations officers on board the Nancy Foster to tell us a little bit about what their role is and why it's important.
My name's Kiana, or Ensign Cadwallader, as I'm known in the service, and I'm the navigation officer on the Nancy Foster. So we're out over the Hudson Canyon, which is a couple hundred miles south of Long Island, New York, and we're dropping an ROV down into the bottom of the canyon to check out what's living down there and to take some biological samples. I work on a NOAA ship, and NOAA ships are captained or driven by NOAA Corps officers, which is what I am. So we're a uniformed service of the United States. We're in the Department of Commerce. And as a navigation officer, my job is to put together route plans for any of the trips that we go out to do or any of the science operations that we're going out to do. It's definitely a journey. It takes a couple months to do the application process to join the NOAA Corps, and it's also a pretty competitive process. So I was in a class of 20 officer candidates, and we had 600 other applicants that we competed against to get into that class. For me, I had previously been working on fish boats, scallop boats, geotechnical boats. I had done a lot of protected species work. And I found out about the NOAA Corps after applying to a NOAA lab and having the lab director refer me in that way. And it just worked out great. This is kind of a beautiful blend of all my passions, right? I really enjoy being at sea. I really love science. So this is an awesome way for me to get to be like operational, out of a desk job, meet a ton of cool people, and learn a lot of new skills. So I've really loved it so far. I consider myself kind of like a science junkie. I'm not a scientist myself, but I do find it super fascinating, and it was my favorite class in school. There's a couple different departments on a NOAA ship. So there's the wardroom. That's the department that I'm in. We're kind of like the ship drivers. There's also the deck department. So they're the guys that you probably saw out on deck helping clip the lines onto the ROV and bring the ROV back on board. There's the engineering department. They're kind of like the secret kings of the ship, right? They keep everything running, and we really couldn't operate without them. And then we have our survey department that helps really make all of the science operations aboard this ship possible. So we have to be in communication with all those different departments all day in order to make operations happen. But especially operations like an ROV, it takes every department of the ship to put an ROV in the water. Communications start and stop on the bridge, so especially for a bridge watch officer, having really clear and concise communications and really controlling that flow of information is super important.
I really loved getting to hear Kiana's perspective, especially how it blends so much of her interest into one thing. And she kind of gets to do a little bit of everything, which is really cool to hear about her time out here at sea. So now I'm joined here with Officer Kate Brogan, who's going to talk a little bit about how an expedition like this can come together and how she supports that mission. Welcome, Kate.
Thanks for having me, Kate.
Yeah, we have two Kates here today. So Kate, why don't you tell us a little bit about what your title means in your position on ship?
Sure. I'm Lieutenant Kate Brogan. I'm the permanent operations officer on board. So that means I'm the liaison between what the science party wants to get done, their objectives and goals, and how the ship can support that, how we can execute those missions. However, on this cruise, I've actually fleeted up. I'm promoted to an acting executive officer, which is the second person in command on NOAA ships. And so my overall job is safety. I'm the direct supervisor of most people on board, and my primary job is the safety of the people, the safety of the mission. I stand bridge watch, so I also operate the ship, and I also do safety officer on the back deck, which means I'm just watching all the operations happen and making sure that everyone's safe in that capacity. And then executive officer does a lot of behind the scenes work. It's not the flashy operations that a lot of you all have seen on these cool videos. It's time and attendance. It's getting people in the right places, making sure we have food, making sure we have deckhands, engineers, officers, making sure that everything goes off smoothly and is supported in the background to make sure the mission goes successfully.
Super cool and really important work. So what's it like having a different science party come on this ship all the time?
It's great. I have a hydrographic survey background, so my first sea tour actually didn't have scientists mobilizing every 2 weeks. So it's definitely different, but it's very cool. It's awesome to see the energy that each group brings and how excited they get about the science that they're doing. I know some people get really excited about corals, some people get really excited about water, mud, all sorts of different stuff. So it's cool to learn from them and then be able to support their objectives as much as we can, minus anything mechanically or with the weather that we can't control.
Yeah, and this cruise specifically are definitely some coral fans and mud fans as well. So how important would you say teamwork is with your crew and also with the science team?
Oh, it's of utmost importance. Teamwork and communication. Communication is really how we make the orchestra play, right? Every single person has a very important role in order to make the mission successful, and one is not more important than the other. So teamwork is absolute, and we always, as ops, actually, I will do pre-project meetings, pre-project planning to ensure that once the science party gets here, that the crew is all on the same page of what we're doing, where we're doing it, and why. So it's really cool experience to be able to see the operations side and then also kind of the other side of the house, the supporting executive officer role as well.
Yeah, and we really appreciate the work and the communication that you've given to us just along the past 2 weeks as well. So one last question I have for you. What would you say is your favorite moment from this expedition over the past 2 weeks?
Ooh, that's hard to pick my favorite. The ROV ops are very cool. We're going to depths 2,000 to 3,000 meters deep. I mean, that can be miles below the surface of the water, which is really neat, going to different monuments and the Hudson Canyon and learning more about it. However, we had this really cool event last night. I was on watch and driving the ship, and there was a spout. So a water spout came up from a whale, and you know how the water comes out from their blowhole. And so I saw one of those, and then I saw a few of those, and then I saw a lot of those. So the ship ended up coming in all stop, and we were probably surrounded by at least 100 pilot whales, including some calves, including some baby whales. So that was really cool experience for everybody on board. We made an announcement. Everybody came up and took a look at all of the whales. We had to stop operations because it's not safe to drive through a pod like that. But once they moseyed on, we were able to keep going to our survey operations. That was a pretty cool experience. And ironically, it was at sunset. So that was also pretty cool.
Yeah, and it was also really cool to be a part of it. And I'll just say the announcement that she made was really funny. It was just like, hello, everyone. We are fully surrounded by whales. And everyone came out, and it was just such a great moment of camaraderie to really just wrap all of the work that we've been doing the past 2 weeks. And it was just something really excited and something that it's just hard to explain how impactful it was, but it was. Thank you so much, Kate. It was really nice to hear from your perspective and what it's like to work on the operations side of things. So a huge part of this research cruise was to collect samples. And so we've shown you how the ROV works down below. We've shown you how it collects samples from the sea floor, but what we haven't really had a chance to show you is what happens in the lab. So we're actually going to meet one of our coral scientists, Dr. Jen Salerno, and she's going to walk us through that process. And then we're going to go down into the wet lab and explore the discoveries from today.
So this was an early interest as a kid. I, like many marine biologists, aspiring marine scientists, grew up in the landlocked state of Pennsylvania. But of course, during the summers, we go down the shore, and we lived in Virginia for a period of time. And this is what I love about this, the outreach that you're doing, because it was through a summer program for students where we got to participate in marine science, where I really caught the bug to do this. And I remember a neighbor asked my best friend and I, what do you want to be when you grow up? The most horrible question ever for a kid, but and she had her answer, and I said, well, I don't know. And then when I found out that I could actually do this as a job, I was set. My name's Jen Salerno, and I am a marine microbiologist and a coral reef ecologist. What that means is I'm interested in studying microorganisms that live in close association with corals, particularly symbiotic interactions. So that means that the coral and the microbes are living in really close association and a partnership for a majority of their lifespan. So I use different tools to do that. I use molecular biology, so that's the stuff you see on like CSI crime scene shows, when you see like the DNA fingerprinting. And another tool I use is microscopy, and that allows us to visualize. So we take a coral and we slice it up and put it under a microscope, and we can actually see inside and see where the microbes are living inside the body of the coral. And that will tell us a lot about what it might be doing there, whether it's helpful or if it might be harmful, like a disease or a pathogen causing organism. Microbes are everywhere. They're all around us, they're in us, they're on us. They perform really important functions. Most people associate them with negative things like disease, going through the pandemic, but the majority of them are actually doing beneficial things for your body and the environment. And they're just really fascinating because they're able to basically eat anything, right? They can metabolize or break down many different substances, including pollutants and things like that. So they have a lot of beneficial uses. Deep water corals live in the absence of light. So what I'm really interested in is what sort of partnerships they might form with microorganisms that allow them to thrive in what we would consider extreme environments. So unlike shallow water corals, they rely more heavily on predation. So capturing tiny prey, tiny animals called zooplankton from the water column and eating them. So coming out to sea, you have to be really flexible. There are a lot of knowns and unknowns. So the things I control are the types of equipment I need to bring out here to do my work. And so I have to really think about my limitations in terms of how many samples we might collect or how many corals we may or may not find. It could be anywhere from 0 to thousands of samples, and you just don't know. So you really have to try to mentally and logistically prepare for that, which can be challenging, but also kind of exciting to think creatively about how you might be able to use different tools or different approaches to do that. And I tell my microbiology students, I had no interest in studying microbiology. I think I didn't have the curiosity or creativity at the time, and now I think it's the most fascinating thing to study.
All right, as you can see, location has changed. We are down and live in the wet lab. So just to share some updates, there's a lot going on right now. If you caught the live feed earlier this morning, we discovered a coral garden. So we have a lot of happy scientists behind me. I'm going to actually share some raw footage from this morning of the reactions in the ROV van. Truly incredible.
Tell us what just happened.
We found one of the larger corals, Aragorgia here, which we've seen in other canyons. It's huge, it's old, it's beautiful. What more can you ask for? It was hoped for, but unexpected, at least until now. This is our last dive here, dive 7, and this is the first time we've seen them. Even though we looked high and low and low and high. This appears to me to be what we call a coral garden. What we've been looking for. Many species, several species, some quite large specimens, covering a wall, essentially.
I mean, if you want to try for a cut, I would say if it's not coming off.
It's a team effort. We didn't know about this last dive. This is a short dive, too.
That behind it is an encrusting.
Icing on the cake.
Like stolen infrared coral, probably.
Nope.
I was like, that makes diving this morning A++, 100% worth it.
We did it.
Yeah. That is a massive tree coral, Primnoa rosetiformis. This one is one that is fish habitat for sure. I'm really surprised to see it here. It's well known from the Gulf of Maine. I don't know how often it's been documented this far south.
That Primnoa, this orange coral, also known as a tree coral, this is the first time we've seen it in Hudson Canyon.
Did you just look that up, or you just know that from your?
I know that from my research on what was here before we came, basically.
Yeah.
And other expeditions to Hudson Canyon. Yeah, I mean, it's not uncommon in other areas like in the Gulf of Maine, but it could be genetically distinct between the two areas, for example.
505, B5, and we've got 1305.
Yes, of course.
I got so excited. Like, there's so much, like when you see a big Primnoa like that in a spot that you're not expecting it, and it's such a distinctive coral that it's just like instantly, that's a Primnoa right there. And you're like, it's not supposed to be here. This is amazing. In this area, I would certainly call this a coral garden. You get into that, you suddenly have species you haven't been seeing. I'm sure there are species in there that we did not see that we'll pick up in the eDNA. That is definitely a coral garden.
So as you can see, really exciting things, lots of tears of joy, just feeling very accomplished and just having really good emotions right now. So let's actually check out behind me. Let's take a look at some of those coral samples, see what's going on. All right, so we've got Meredith. She's opening the fridge.
Our first find of the day, we came around the corner, and this is Paragorgia arborea. So Paragorgia arborea, we were not expecting to see this, and when we found it, we're like, we got to get a piece of that. So here it goes. I'm going to take it over to the table here behind us for processing.
Oh, wait, is that big?
It's that big.
Pull it out.
Okay, want to grab it out?
That's skate eggs.
I'll get our
Oh, it's beautiful.
water tray out of the way.
So we got Dave Packer here. This was the Holy Grail, is what you called it.
The Holy Grail of corals. Well, when you're looking for corals, you want something big, bold, and beautiful, and that fulfills it. And also what's interesting is these are skate eggs, mermaid purses, that probably a skate, maybe not a shark, laid them on there. So I don't know if we have to raise them to maturity and keep them as pets or what they're supposed to do with it. But this just happens to be one of the corals that we like to see because it's found in other canyons. It gets big, it's old, it's got 3 color morphs, red, pink, and white. And you just know you're getting good corals when you see these big colonies like that. And this is just a piece of it. I mean, it's hard to tell on camera, but they can get pretty big, actually. And so I think, Jen, you're next on this.
So what am I need over here? And this is considered a hard coral?
Well, technically it's a soft coral, but it's related to gorgonians and the kinds that you would see in tropical reefs.
Yeah.
So.
So now Dr. Jen, who we met in the video, she's now here live. Welcome, Jen.
Hi. So I think Dave is preserving this as a type specimen for the museum, right? For vouchers. And I'm taking little biopsies to look at the microbes that are associated with the coral, both externally, and I'm really interested in those that might be found inside. So I'll take one little snip for microbiology. So I'll preserve it in some DNA RNA buffer, and the other one I'll put in preservative for microscopy for later. So, and I've never dissected this one. So like I have a bunch of different tools because you never know what you're getting into. Some of them have hard skeletons. These are soft corals, so they should be easier to cut through, but I say that, you never know. Okay, that wasn't too bad. It's kind of spongy. This one, the common name is bubblegum coral, and you can see from the coloration and the morphology why it's called that. So I'll take a couple snips. I don't need a lot of tissue, and I try to get representative tissue from around the coral. And I think I'm good, so you can cut the rest of that.
Yes.
Thanks.
That's why, again, that even though she called it a soft coral, there are soft corals and softer corals. This is more towards the hard end.
Okay.
Not stony, but close enough. And now all I have to do is figure out where the heck I'm going to put this because I don't have a jar big enough.
I lied. I'm going to take one more. Just going to take one more.
Oh, great.
Okay.
Jen's going to take another piece. Taking another one. It's so interesting that you only have to get the tiniest sample,
Yeah.
but it can tell you a lot.
Yes. This might not be that graceful. Sometimes you're just kind of. Yeah, you know what? I think maybe the scalpel might be good for this piece.
Scalpel will also give you a nice cross section where you can see the inside,
Yeah.
which has the diagnostic character.
Sure, and you can see like all these little, I can see the nubbins are where the polyps are. So the polyps you can think of as corals are colonial organisms. And so if you think of an anemone, how it's a mouth surrounded by tentacles, if you shrink that down really small and essentially have a bunch of really tiny anemones and put them across a coral colony, that's what a coral is. And in many cases, their what would be their stomach or their gastrovascular cavity is connected. So they're all connected in there. So there's many, many polyps in this one coral colony. Okay, thanks.
So what are you going to do now, Dave?
So I think that it's so large, I don't have a single bottle, so I'm probably going to have to divide this up and put them in two and then label them as such. But the main thing is that these are going to the Smithsonian Institution as what they call voucher specimens, which means everything goes with it, including the skate eggs. And they'll be curated appropriately and identified and we'll check our taxonomy, basically. So we're sure we're on the right track. We're pretty sure, but, coral taxonomy is kind of a mess because the names keep changing all the time and scientists are discovering new species all the time. Sometimes the old species become the new species. Sometimes it's an actual new species. So we want to have a good collection at the Smithsonian, and we have folks there that are specialized in deep sea corals, and so they can do what they need to do in order to identify them.
That's great. Yeah, it's really cool knowing that the research you're doing on here is going to go to so many different places to help better it that way as well.
Yeah.
Why don't we hear from our chief scientist, Meredith? We've got a lot of firsts happening on this cruise today. So I'd like to hear from Meredith on her first time being a chief scientist.
It is my first time being a chief scientist. This has been actually a great cruise to be on as a first-time chief scientist. The crew here has all worked really well together. There's a great team to work with, both the ROV team, working with Layla from USM and OECI has been fantastic. I've gotten to work with Dave remotely before, but always online, so it's been great to work with him in person. Really fantastic cruise. I've had a really great time at it. So glad to come out here and do my first East Coast cruise too. First cruise in the Northeast.
Oh yeah, amazing. You're from the Northwest,
Yeah.
so this is really cool.
Yeah.
Slam dunk, I would say, for a final day.
Yeah, like this was didn't go in today expecting much, and was like, I'm so glad we just went for that final dive. This was like the most spectacular dive of the whole cruise.
Yeah, I agree. Well, speaking of another first, Adrian, if you don't mind me. So you said this was your first time on an expedition like this before. How do you feel?
Oh, it's been amazing. This is obviously my first time being out on a cruise, and I think just seeing all the expertises and all the different people coming together to really make something like this happen has just been spectacular. Get to meet so many people, I have so many questions, and it just getting to really experience everything and how something like this comes together with all these different expertises. Given my coral expertise, I feel as though in academia you're always often so siloed, and to see these ROV engineers, these survey techs, everyone sort of coming to the bridge, come together to make a mission like this work is just amazing.
Well, I think people seeing you out here as a student is inspiring a lot of younger people, maybe who are watching in the audience as well. So the work that you've done in the past 2 weeks, I think, is definitely incredible, and it's going to bring a lot of joy to a lot of people.
Thank you.
So chat and audience, why don't we answer some questions and see what people are wondering about? Let's Yeah, feel free to throw any questions in the chat, anything that you're seeing. I know it's like there's a lot happening right now, but this is all happening live and in real time. Like I said, the ROV just came up maybe 15 minutes ago, so this excitement and this type of joy is really just it's all in real time. So it's really cool to get to see everybody's reactions of this coming out. And even while we were upstairs, when we were looking out into the water, the team down here, they waited to bring out the corals until we knew that the audience was with us. So by the time we got down here, Meredith's like, okay, I'm ready. Can I open it now? Can I open it now? And I'm like, hold on, just a second. We want to make sure everyone gets to see it. So we saw that in real time with you all. So let's see, we've got a first question. Okay, let's see. All right, so we've got a question. How many species of corals have been documented living in the Hudson Canyon? Anyone familiar?
I'm going to just guesstimate. It's only been like 10.
Okay. Only about 10.
That sounds about right.
Yeah.
There's a great place that people could go and find out. NOAA's Deep Sea Coral Program runs a national database of deep sea corals where they have actually a mapping tool where you can visualize their locations and actually zoom in. They're starting to get photographs in there. So if someone wanted to go like do the research and check that out for themselves, you can actually zoom in on the map in Hudson Canyon, and there are some tools there that will give you species counts, what the densities are from any surveys that have been previously known. So yeah, check out the Deep Sea Coral database, and that's the kind of place where anyone in the public could find out that information.
And this information will be going into the database, basically, because if you do look at Hudson Canyon corals in the past, you will not find very many on the maps. So this will add to it.
Cool to know that this is going to be inputted into the database, and we're all seeing it first. Okay, let's see. Oh, I think this is a question for our mapping specialist, Tori. Tori, let's see. Someone said, I'm curious about the success rate of finding coral habitat that was predicted by the models.
That's a good question. So the models didn't actually use data here in the canyon. So they're using data from outside this canyon. So we really were kind of guessing. I mean, the models can make predictions, but if it doesn't have data here, then it's kind of an extrapolation in some way. So now what we can do, now that we have all this great data, we had some toes where we found coral, some where we found mostly mud. So we can put that into the models and then get more results out. Is that good? Anything else to follow up?
I don't think so. That was cool. Thank you, Tori. Let's see. Got some ROV footage. This is just the ROV working to collect some samples again, like we've seen throughout time. Okay, let's see. Someone said, hey, Kate, what has been your favorite moment from the cruise? There's been so many things that have been really exciting, but I think for me, we got to experience bioluminescence at nighttime, which was really cool and something I wasn't expecting. And it wasn't like a ton of it, but you could just see it kind of rolling up. Oh, yeah, we got a footage of that too. It was just such a magical experience getting to see that. And then if you looked up, you're staring at the Milky Way. So I don't know how it could get much better than that. So that was probably like top moment for me. Also, the pilot whales that we were talking about earlier, that was incredible. And also the coral discoveries, super cool. And I just like getting to be a part of so many firsts for so many things on this cruise. Looks like this question might be for Layla. For driving the ROV, are there simulators or training dives in ROV driving test?
That's a great question. There are, and it really depends on what type of ROVs that you're driving. If it's if you're driving it for the industry, there are really strict rules that you have to check out with the pilot training. But this is actually something that the NOAA Ocean Exploration Cooperative Institute is trying to open up as a career path for students in all of the universities that are within the OECI, of how can we get students and ocean engineers and
involved in not just the build of the ROVs, but how we fly them around. And we expect within a year from now that we're going to have some progress with pilot training programs that are offered directly through the OECI and supported by NOAA. You're kind of at a moment where what we need to do to get people able to drive ROVs is going to change, and we're really trying to open up that as a career path and a field for a lot of the students that we work with.
Super cool. And also, I see that you're next to a big hunk of mud. What are you doing?
Yeah, so we've been sampling the base of the canyon throughout the entire cruise. I'm a microbial ecologist where we think that the history of the ocean is written in the mud. And so by sampling down core every couple of centimeters, we're getting little micro profiles of how the organisms got there, what kind of distribution that they have across the seafloor. This one is kind of challenging, though. If you look in the middle here, if anybody from my lab is looking, they're going to think this is very messy, but there is a rather large critter that's right dead on center that's taking up most of the space of the core. And I could spin it around. You can see it's kind of frilly over here. I think it's actually more than one organism. This might be a brittle star that we cored through. This might be a tube worm, and so I'm basically excavating this core as I go down. I'm sort of making it up as we go along, since so much here is actually critter and not mud. So we're sampling around it, and I'm just peeling this down layer by layer until we see what we have captured in this moment in time in the mud.
Super cool. Thank you, Layla. Yeah. Well, I guess speaking of creepy critters, we actually saw something really cool and a little spooky. So we kind of took a screen grab of this and put together a video of this can't be missed highlight. So let's check it out. So these are what we think are a type of worm. And so you can see them moving like that. And they're very scary looking. And we stumbled upon this and we were like, oh my gosh, we have to take a closer look at these worms. And we think these are a type of spoon worm. Stuff of nightmares, but really cool. So I'm glad that we get to show you some of the creepier things that we've been seeing as well. Let's check out and see if there's any more things in the chat. So I think what we'll do is we're going to end with one final question, and we have a few other things to show you as well. But this question is going to be for Meredith and anyone else who wants to join in. So Meredith, how does today and your findings change your perspective moving forward?
What I had always heard was that it was just very muddy and we weren't sure what we were going to find. And today, it's definitely got coral habitat in it. And I'm really excited to see how the data we were able to collect today is going to feed into new species models and things where we might be able to better predict where we can find corals in this habitat and just understand more about the overall biodiversity here. That's the kind of stuff I really love. I love doing this kind of work that's kind of science in the public interest, where we can preserve habitats for generations before and also, preserve them for groups like, fishing communities and things, because, it's all connected. So healthy habitats like this lead to healthy fisheries.
Yeah, I agree with that. And it's so cool getting to learn more about Hudson Canyon, as this isn't a very actively researched or looked at place. And now it is becoming more and more interesting. So it's really great that we get to share that with the public and more people can understand the super cool science behind what we do out here and what's been happening in the canyon. So now I know I've showed you, we've learned a lot from the science party behind us. We've seen a lot of different things throughout the past couple streams. But what I really want to show you all is like a really fun best hits of everything that we've seen on the live streams. So if you haven't been able to tune in to those 9-hour long dive days, we put together some really cool creatures that we saw, some cool corals, and just some cool footage that was happening down below in the deep. So let's take a break and take a look. Wow, what an amazing journey. We've seen deep sea species, explored hidden ecosystems, and witnessed the dedication of our incredible crew and scientists. This expedition may be ending, but the discoveries will continue to impact ocean science for years to come. So thank you to everyone who joined us live, and don't forget to follow Noah's work and stay connected to the next adventures in ocean exploration. Until next time, keep exploring and stay curious, and thank you so much for tuning in on Hudson Canyon Live.